Hello, welcome to the National World War II Museum student webinar. Um, Today we're going to talk about innovations and treatments and medical treatments, and particularly vaccines in World War II. A few reminders, if you have questions or comments, put them in the Q&A window. We've got the chat window closed, and this will be recorded and will be posted on YouTube and Facebook for you to use later. After it posts, I'm going to put on our teacher Facebook page links to some documents and some lessons that will help you out. So please look there, join our teacher Facebook page if you haven't already, so that you can find those resources. We have some hands-on activities you could do related to it and some computer simulations about epidemics. And with that, we're gonna go right on in. Can we go to the slides, please? So these are pictures from 1937. And you see something kind of like we see today. We see mostly empty classrooms in some places. We see students working from the latest media technology. In case you don't know, that big piece of furniture behind those kids is a radio. That's how big radios were back then. And so why do we see that? Well, in 1937, there was an outbreak of polio, and people were really scared. So in places like Chicago, they kept their kids home and students worked from home. They listened to the radio to get their lessons. These are some newspaper clippings from the same time. So you could see these in 1937, these students stayed home for a while, and they had a radio schedule so that different classes were at different times, and they would gather in there and see what was going on in their classes and learn from their teachers. So polio, what was polio? Polio was a really frightening disease. It really scared people. One big difference between it and our current pandemic is that it affected children, mostly children. So polio was a virus and kids could get it, and it would cause paralysis. Sometimes people would recover, and sometimes it would just keep getting worse and worse. Some people died. Some people were permanently paralyzed. So here we have a picture of a girl. This was a comment site. I remember my third grade teacher was like this girl. She grew up. She obviously wasn't a little girl when she was my third grade teacher, but she had had polio and she wore a brace and used crutches for her whole life. This is a summer camp where they did rehabilitation for polio victims. And if you look in the center, there's an older man in there, and that is our president, who was president during World War II, Franklin Roosevelt. And here's a picture of Roosevelt too. So Roosevelt was an unusual case. He got polio when he was an adult, and it got pretty bad. He could not walk very well on his own, and he wanted to project an image of strength, so he rarely allowed people to see him in a wheelchair. This was at home with the daughter of his groundskeeper and their pet dog. I love that picture. So parents were really afraid of polio because it could get this bad. These are called iron lungs. This is a hospital ward where all sorts of polio victims are in these iron lungs. Their polio had gotten so bad, it attacked their muscles, even the muscles involved in breathing couldn't work. And so they had to have they were in these machines that had different amounts of pressure in them coming in and out so that it would expand and contract their torso so that they could breathe. That way they didn't have to be on a respirator. They could talk and everything, but it breathed for them. So this was really terrifying. And it went all the way until in the early 1950s, this gentleman, Jonas Salk, led a team that developed a vaccine for polio. And it was huge for the world because no longer did people have to fear this disease for their kids. So how did this all happen? Well, it was really it was really difficult to come up with all this technology because until 19 the early 1930s, they weren't even really sure what a virus was. Now they knew they could put it through a filter so that it was much smaller than a bacterium, but they weren't sure really, was it a living thing? Was it a protein? Was it just a bunch of strange chemicals? They weren't sure what they were until some German scientists in the early 1930s built an electron microscope that could visualize viruses so that when they filtered out um, the the solutions that had the disease causing things in it, they could find the viruses. And then later in the 1930s, they discovered these are the polio vaccines, and a little sign that's encouraging you to take the polio vaccine. By later in the 50s, they had an oral solution that you could take. Um, so these are scientists doing something strange with eggs. The only way that they could grow viruses was to put them inside of an egg. Now, viruses, this is really tricky. Viruses are not really alive. Living things take energy from their environment. They take chemicals or light or something, and they make energy that so that they can build more of themselves and reproduce. Viruses don't capture energy. They don't generate energy. They're complete parasites. They get inside your cell and they get your cell to make copies of themselves. Um, and it's a really funny way to go. So it's hard to think of it that way, but they really aren't alive. So that you can't grow them in a culture dish with food like you would a bacteria because they don't need food. They have to be inside of cells to survive. So they would put them inside these eggs, and then the eggs would be infected and lots of viruses would grow. And then they would take them back out of the eggs and separate them from the egg material. And that was the only way they could grow them. And so it took a long time to grow lots of eggs, to grow these viruses, and it took a lot of eggs and a lot of work. But they were able to eventually grow them and find some that they could inactivate so that they could put them inside our bodies, and our body would have an immune response to them, but they wouldn't make us sick. And so that's how they developed the polio vaccine. Now, this guy, Jonas Salk, who developed this, he had earlier worked in the 10 years before or so on another project to develop the first flu vaccine. So let's go back further in time and let's talk about the 1918 flu epidemic. So sometimes they called this at the time the Spanish flu. To be more scientifically correct, we don't refer to the place where we first found it. We refer to the timing of the virus or something else because we aren't really sure where it came from. The 1918 flu actually may have originated in the Midwest of the United States because there's a lot of evidence that supports that idea. So anyway, in 1918, there was a huge flu pandemic. So a pandemic is across the whole world. That means everybody across the whole world. Pan means all over the place, okay? And so that's what a pandemic is. And this was a pandemic. Our current coronavirus outbreak is a pandemic because it's affecting the whole world. An epidemic is if it's just really bad in some places, okay? So polio had a whole bunch of epidemics, but it never became a pandemic. It was never one worldwide event. So this pandemic in 1918 was really bad. It was, in terms of the population size of the world, it was worse than our current pandemic. Many, many people died. There was lots of disruption in the world. And so you could see in this newspaper, they closed schools and theaters. Now, interestingly, I showed you a photo earlier of students staying home because of the polio epidemic in 1937 in Chicago. Well, some cities like Chicago and Washington, D.C., did not close their schools during the 1918 pandemic. And those places ended up having some of the worst outbreaks and the highest number of deaths. So maybe closing schools would have been a good idea. So these are some high school students from 1918 wearing masks. You can see that people wore masks back then. You can kind of see their faces through them because they used gauze masks. They didn't really know what a virus was, so they didn't have really great masks. And lots of other disastrous things were going on in the world at that time, kind of like now. Um, so there was a war going on. World War I was at the same time. Actually, the worst month of the 1918 pandemic was when the war ended in November of 1918. That was the worst month of deaths. So all these soldiers moving around, even if they wore masks, probably was not helpful to containing this pandemic. So this was caused by a virus. But remember I said they discovered what viruses were in 1930s. So in 1918, they had no idea what was causing this. So it was really very, very scary. They had basically no treatments and no idea what was causing this. But by the 1940s, by 1944, we had developed a vaccine for it. The military and the rest of the government really invested a lot during World War II in trying to develop a flu vaccine because they were worried about the same thing. It was a time when lots of people were moving around because of people that were displaced because of the war, soldiers that were coming back and forth across the world. So they were really interested in developing a vaccine. So they, in the 1930s, one of the first vaccines they started studying, besides polio, was the flu vaccine. And so by 1944, they had tested a flu vaccine on soldiers, on army men, and found that it worked pretty well. And the first few seasons, the flu vaccine worked very well. And then it pretty much failed in 1948. And that's when they discovered that there's more than just one kind of flu. There are different strains of flu. So the flu is a vaccine where we take, because the viruses differ a little bit, we take a vaccine that's they call it a polyvalent vaccine. That means it has many different strains of viruses in it, a few different strains, and that way we're protected from whatever comes out. Because when they make it in the summer, we aren't sure what strains are going to be around in the fall or winter. So they're constantly looking at the environment around us, what viruses are in animals that will change to us, because that's typically where these new viruses come from, is they come to humans from animals, especially from pigs and birds. And so they keep track of populations of wild birds and domesticated birds and pigs and see what viruses are in them, and they project ahead to pick which strains to put in the flu vaccine. So let's switch to talking about today. So in the years since the polio vaccine and the flu vaccine, scientists have developed many, many different vaccines. And you kids today are not going to get many diseases that people like me got when we were a kid. I got the mumps when I was a kid, and it was terrible. And many people can have bad complications from the mumps. You will never get chickenpox probably because you have a chickenpox vaccine, and the chickenpox is a really bad one because it can come back again when you're an adult and cause a disease called shingles. So if you look at this is a chart, and the yellow vaccines are the ones that pretty much they really want everybody to take, okay? And so you are protected from all these different things, including a couple of different kinds of pneumonia, measles, mumps, rubella, a different kind of influenza, chickenpox, hepatitis. These are all in different vaccines that you get as you get older. And so vaccines are really great. They have kept millions of people alive who probably would have died from these diseases otherwise. So how do we develop vaccines? So what we have to do is we have to understand how the body works. And of course, we know much more about this today than we knew back in World War I or World War II. And what we know today is that when a foreign agent like a bacteria or a virus comes into your body, like for example, the chickenpox. So you can look at that slide and look at the chickenpox. So it comes in and your body needs to develop an antibody to attack and destroy that virus to keep it from making you sick. So normally, if you look at the green one where it says new pathogen, you'll see that when a new thing comes into your body, your immune system, if it has never been exposed to it, has no antibody for it. And so you can get pretty sick as it reproduces in your body until your body can develop antibodies for it. Now, people discovered almost 1,000 years ago that we can prepare our bodies for it. And that was with smallpox, was they found that people who, and Europeans discovered this, but lots of other people discovered it too, that people who had been exposed to related diseases in livestock, like cattle have their own cowpox disease, that those people who had been exposed to that didn't get sick or didn't get as sick as people who hadn't been exposed to that disease. And so sometimes they would take blood or the infections from the little measles spots on the animals or on people who are sick, and they would put them under your skin so that you would get really sick. Sounds gross, doesn't it? But it was, they were desperate not to die from smallpox. And so that was how they did it. So that was their first attempt at vaccines. Today, we can be much more strategic. And so what we could do is we can give your body an advance notice of what's coming. And so what we do with a vaccine is that we give just a part of the foreign agent, the bacteria, or usually a virus, okay? We either weaken it or we give you just a part of the virus so that your body can develop antibodies for it. Like, for example, last night, I'm very excited that I got my first coronavirus vaccine. And so my arm is a little sore. And that's because my body is responding to a little bit of the virus. Okay. And so there's ways that we can do this. So we don't have to give the whole disease causing organism because that might make you really sick. It also means if we're trying to totally get rid of this virus in the world, like they're totally trying to get rid of smallpox and polio, like if they give it to you, that means that there's still some of it around in the world. So we want to give a weakened or just a tiny part of the virus to our bodies so that our body is ready to attack it. So there's 3 ways we could do this. We can either give you the whole virus or bacteria, or we can give you just some of the parts that trigger the immune system. Now, with a virus like the coronavirus, they have these sticky things on the outside of them. And these sticky things are what allow them to attach to a cell and get pulled inside the cell where they get the cell to make copies of themselves, like little pirates sneaking inside your cell. So if we can give your body just the sticky parts, then your body will know how to attack it, but the virus won't be able to replicate itself. So we could either give you just those little proteins, or this is a new trick that they've just discovered how to do. We can give you just the genetic material that makes those sticky parts. And that's what's in the current vaccines that are available. All 3 of them, the Moderna vaccine, the Pfizer vaccine that I got, and the Johnson & Johnson vaccine, all 3 of them use just the genes that make the sticky protein. They just give you that, and then it goes inside your body. Some of your cells start to make that sticky protein, and your immune system responds, and so you may feel a little sick, a little bit sore, and then but while your immune system is responding to this new thing that's in you that probably shouldn't be in you, and then it's ready. And they give it to you twice because, except for the Johnson & Johnson one, because it takes a little bit of time for your body to remember. And so it has to be a big enough event that your body remembers and keeps making these antibodies, because it takes a while to generate these antibodies. So this is how these vaccines work. And over all this time, many scientists have done this. But think about how big an effort this is, not just to develop the vaccine, but then to produce all these copies of it. So they have to make all those copies and all that vaccine and replicate all that DNA over and over again, and then find a solution to put it in. And then they have to make all those little jars and all those millions and millions of syringes. So think about it. In this country alone, there are about 300, almost 350 million people. Now, if everybody's going to get 2 shots, that means we have to make 700 million syringes. And that is not an insignificant task. I mean, we've seen over the last year that, we ran out of flour at the grocery stores and mason jars and things like that. So if we're going to run out of that, imagine how likely we are to run out of specialized materials. So we're doing today like we did in World War II, and we're asking companies to go ahead and help to make stuff that they might not otherwise make to support the effort. And so some companies are that haven't developed a vaccine of their own, like Merck is making more of the Johnson & Johnson vaccine so that they can support the vaccination effort. And all sorts of pharmacies and stuff that normally are not getting involved in giving vaccines are delivering them. So we can look back when we face something like we have this big pandemic today. It's been scary. It's felt like this is the worst thing that's ever happened in the entire world. But when we look back at history, there have been other times where people have looked and said, this is the worst thing that's happened in the entire world. And we can look back at how people survived those times and use those strategies and those ideas to help us today. All right, so that's the last slide. And we can go on back to the camera and I can take some questions. So I'm gonna give you, while you're putting in your questions, I'm gonna give you some ideas of what are in the resources. So in the real world science curriculum, we have a lot about the development of penicillin. And one of the lessons in there is about different kinds of cells and what features they have and what we have to do to attack those kinds of cells. That lesson's called antibiotic targets, and I'll put it in there. We also have blood in a bag, which is a lesson that we talked about in my last webinar about blood plasma. And then there are, I'm going to give you links to some online simulations where you can see how epidemics spread, and some activities where you can simulate in your classroom epidemics and how vaccinations can stop epidemics. All right, so let's see if I can read these. Please, okay, some questions. All right, so some people want to know, are viruses really alive? They aren't alive. I know that's confusing to a lot of people, but because they can't live on their own at all, they can't take in any energy, we can't grow them in a dish full of food, we say, no, they're not alive. All right, so email any questions you have, and contact us on the teacher Facebook page, and let us know what else you want to learn about and what help we can give you. Thank you very much for tuning in. We'll be back in a couple weeks to talk about artificial intelligence and the roots of computers in World War II. And until then, this is Rob Wallace at the National World War II Museum, and we'll see you later.